Hello, everybody. My name is Nolan Nichols, and I lead the engineering effort in the data sciences group at Maze Therapeutics. And today I'm going to be telling you a little bit about Maze and how we're leveraging some of the AWS projects in the healthcare and life sciences space. And so to get started, I wanted to tell you about a little bit about Maze and how Maze was founded to answer this fundamental question around why do some people get sick and others don't, even when they have the same disease-causing gene. And so there are two kind of key insights that enable being able to answer this question. And so back in 2016, when our founders were first developing the concept around Maze, a paper was published by the Resilience Project where they identified individuals that should have a serious childhood disease, but they did not. And so we set out to ask the question of why. And so it's broadly known that there are genes that can provide protective effects over certain diseases. But we are curious about if this insight into so-called genetic modifiers could be used as a general platform for identifying therapeutic targets. And so around this same time that this paper was published, two of our founders, Jonathan Weissman and Steve Elledge, were developing novel applications of CRISPR technologies to do genome-wide gene-gene interaction studies. And they were looking into how these tools could be used to kill cancer cells. But the interesting thing is that while Jonathan was looking at these synthetic lethal combinations, he also found that there were protective combinations that his lab described as having buffering effects. And so this further built on the idea behind the Resilience Project that you could identify these protective modifiers. And so what the Maze team wanted to do is see if they could take John and Steve's concepts and then turn those into a drug company. And so the early team at Maze started to build out the company, and they thought that they could identify these naturally occurring protective variants from human genetics data, and then be able to generate proprietary functional genomics data that could be used to validate these protective modifiers and develop new therapies for severe genetically defined diseases. And so our approach has been to develop a purpose-built platform that we call Compass to integrate high-value human genetics data from a variety of biobanks, along with functional genomics data that we generate internally, and then also bring in together commercial data and other proprietary sources to be able to identify new targets and then conduct genome-wide CRISPR screens that can be used to understand the biology underlying these genetic modifiers. And so once we've amassed a critical body of evidence, we can use what we've learned to then focus the drug discovery side of the organization in a more data-driven way. And so now that you have a little bit of context around Maze, so I'd like to switch gears and start to unpack the importance of having this integrated human genetics and functional genomics data by leveraging data science approaches. And so, as you might be familiar with, over the past few years, it's been widely cited that data scientists and data professionals spend a disproportionate amount of their time going into data preparation rather than doing analysis itself. And so in 2020, there was a survey by Anaconda on the State of Data Science report that indicates that data professionals still spend around 45% of their time on data management related tasks. And these data management tasks are really essential to the overall data science lifecycle and provide a foundation for all of the different preparation for analysis, visualization, and other forms of reporting. And so at Maze, what we want to be able to do is provide our data scientists and computational biologists with clean, structured data in the formats that they're familiar with. So using tools like Bioconductor, things like summarized experiments, which capture all of the metadata and data all within a convenient to use API. And also using tools like R Markdown to generate reports. And we want them to be able to focus on that aspect of their work. And so what we've been developing is something that we call the Maze CLI, the Maze command line interface. And what this is basically provides our analysts with a one-stop shop for doing all the relevant bits of work for their analyses. And so everything from creating a new project that has a Git repository where they can start working locally on their machine, to then spinning up an EC2 instance and hopping onto that to run some containerized tooling and submit that to AWS Batch. This is all really packaged up and provides them, at the command line where they spend most of their time, with access to a wide variety of tools and infrastructure. And so this analysis environment was something that was really critical to getting Maze off of the ground. And so when we were first building out Maze, we were able to work collaboratively with the healthcare and life sciences group at AWS to deploy this AWS biotech blueprint. And so initially, we just deployed a single account, which provided support for a few years and allowed us to get, essentially from just the concept of a company to actually having, an analysis environment, following the current best practices, and be able to start working pretty much immediately. But as Maze started to grow and we had additional functions and additional requirements, we started looking at the Biotech Blueprint 2.0, which provides support for multi-account architecture. And so what this enables is having a single master account that can drive basically all of the user access and controls from a single location, be able to centralize a networking within this transit account, and then provide tailored accounts for specific uses. So for a shared informatics account or research accounts, or specifically around the data science environment that I discussed previously. And so one of the areas where this provides the greatest benefit is around this concept of the transit gateway. And so the transit gateway solves this problem where what we were running into in our single account setup is that as we had a variety of different VPCs for production or data science or development workflows, we had we needed to maintain a VPN connection from our on-prem to each of these different VPCs. And then we started needing to have more cross communication from one VPC to another. And so we started setting up all these VPC peering connections that have a variety of rules associated with them. And on expecting the company to continue to grow, the management task around all these VPC connections ended up being quite a burden. And so this was solved by the transit gateway through this interesting piece called the transit gateway, where we're able to have a single connection from on-prem, a VPN connection to the transit gateway. And then you have this hub and spoke model where all your networking is centralized. Accounts can communicate to one another through routing rules that are defined within the transit gateway. And similarly, we have an individual account for our different use cases. So things like within the data sciences account, we're able to limit the blast radius of what people have access to and any kind of damage that could be done. And similarly for external contractors and others, we can have specialized accounts that really limit what they're able to see and work on. So now we have this kind of architecture through the biotech blueprint that provides us with an analysis, a tailored analysis environment, and then now we need to access some data, and a few of the areas that our data comes from is both from are all from vendor data or other shared data sets from public repositories, as well as our internally generated data from our instruments. But we also leverage the AWS registry of open data. And what's really nice about the framework that's set up here is that we're able to register these different sources of data all within a highly structured AWS S3 object storage, but then register these in performant databases like Athena. And for more specific bioinformatics and human genetics analyses, we leverage a platform called SciDB, where we're able to process hundreds of thousands of patient samples very rapidly, all through this high performance database with with all of our genomics assets from different biobanks all in one location. And so on the AWS side, what we were able to leverage is a project called Data Lake as Code. And so this is a framework where you're able to enroll existing data sources. So these can be from the AWS Open Data Registry or from our internal sources, and then use AWS Glue to transform all of those different types of data into cloud native formats like Parquet, where you're able to have very efficient processing. And then these all get registered within a metadata catalog within Glue. So you can search and query and find all the tables and schemas across this entire data lake, and then analyze them using tools like Athena, or plug them into other downstream business intelligence tools. And so this provides really rapid access to a variety of data sets that are on this registry of open data that I'll talk about next. And in this registry, there are currently 73 life science data sets. And what we're able to do is take a few of the example data sets that are leveraging the data lake as code. So these are GTEx, OpenTargets, BindingDB, and Kimball. And by simply deploying a CloudFormation template within our data science account, within about an hour, we were able to have access to over 150 million records of all these biological and chemical entities, as well as like their properties and relationships in these highly performant formats like Parquet, where previously we were running an analysis where we'll use a public API to query, for example, open targets, and the request would take hours or even days to go and comb through a series of queries. And we were able to take that and trim it down to just a few minutes or even just seconds by leveraging Athena. And so there's still a lot of challenges that remain here. So there are tons of tables, over 18,000 unique columns of data. And so we still need to be able to distill out like what the relevant bits are within this data lake, as well as combine it with our internal data in order to make sense out of it. And so when that comes into play, that's really what our analysts are working towards, is being able to go through all these different data sets, identify the relevant bits that are interesting, and then go through this publication process where they're able to publish their results in our semantic data catalog, and then provide those to non-computational users and biologists downstream through drug target dashboards and other forms of decision support. And so along those lines, I'll walk you through what this publication process looks like. So what we have is we define an ontology that has specified terms or relationships, and you can think of this as really the kind of like schema and controlled vocabulary for how we publish our results. And we generate a description of each analysis using something called JSON LD, which allows us to capture a lot of structured tags that enable further downstream processing, so that when an analyst publishes a result, they capture specifically that this is a differential expression result that uses ensemble IDs, etc. And so you end up having this structured format that we can then validate using the shape constraint language, which is called Shackle, so that we know that only clean data is making it into our publication portal. And then we have this validated data set description that gets published into the data portal. And then we have that available through both a search index for the metadata, and the underlying data sets themselves are served over a data API. And so just in summary, so what we were able to do is rapidly deploy our entire data architecture just over a few days, rather than the months or longer that it would take to get started. And what this provided us with are a number of reference data sets, as well as the reference architecture that we could start from and start tailoring that to our own needs by starting from examples and actual working code. There are still barriers in order to weave these public data together with our own, but our approach has been to leverage AWS and these other open architectures, and start thinking about how we can fill in the gaps, moving from target discovery and validation into the drug discovery space by leveraging these cloud native data resources. And so there's still a key gap to fill here that we're currently working on, where we really want to understand the semantics and the links between the variants and drug targets, and understanding the biology and how those can be linked to compounds that are most relevant to drug discovery into patients. And so with that, I'd like to thank you for your time.